Good morning, everyone.
Morning.
Good morning. I think we can go. So thank you, Stephanie. I am Barbara Lavery, your moderator for the Reimagining Cancer Treatment panel. I'm the Chief Program Officer of the Alliance for Cancer Gene Therapy. We're a research-driven nonprofit based in Connecticut, but I'm out here in LA. And for the last 20 years, we've been funding academic research, specifically in cancer cell and gene therapy. Our founder had the good luck and the prescience to meet Carl June 20 years ago. And when nobody else did, believed in his work in CAR T therapy, which has led to 3 approved products now that are truly saving patients' lives with blood cancer. So we've continued our funding, and now we're focused on solid tumors, hoping to bring that same reimagination of what cancer treatment could be to patients with cancers like pancreatic cancer or glioblastoma, who really have very little options today. So that's me. I'd like to introduce our panelists for today. I'll quickly introduce and then let each of the panelists give you an idea of how they are reimagining cancer treatment and using artificial intelligence, machine learning, and all kinds of things that I know very little about, but will help the discussion today, I hope. So first up, we have John Desjarlais. John is the Chief Scientific Officer at ZenCore. We have Stephanie Culler, the co-founder and CEO of Persephone Biosciences. We have Adamyala, a PhD candidate at MIT CSAIL, and Zora Shams, who's joining us from Cambridge University, a senior researcher in the Department of Computer Science and Technology at Cambridge in the UK. So John, let's start with you. If you just want to give us an overview of what you do at ZenCore, and then we can move through introductions.
Yeah, sure. Hi, John Desjarlais. Again, I'm the CSO at ZenCore. ZenCore is actually an LA area company in Monrovia, California. We've been around since the late 90s and started working on antibody therapies in the early 2000s and started working on immunotherapy using our antibody platform sometime around, basically around 2013, around the same time that we went public. So we specialize in bispecific antibodies. That's a kind of a newer technology in the antibody space where you can interact with 2 different targets at the same time, and we use those to recruit immune cells to attack cancer cells, and are developing treatments for various either blood cancers or solid tumors.
Thanks, John. Stephanie, why don't we have you go next?
Great. Hi, everyone. I'm Stephanie Culler. I'm the co-founder and CEO of Persephone Biosciences. At Persephone, our focus is in using real-world patient data, augmented intelligence, and synthetic biology to develop equitable precision medicine in oncology for the gut microbiome. And towards that, we have a technology platform that essentially has 3 components to it: real-world patient data. So we essentially acquire thousands of patient stool samples reflecting the gut microbiome, blood samples reflecting the immune system to map out the gut immune axis. We want to understand, how does the gut microbiome impact the immune system and therefore disease? We apply a suite of multi-omics technologies to decode that novel biology, and we use augmented intelligence, which is essentially just a combination of artificial intelligence with our own rational intelligence to understand this biology and create a deep knowledge base. And it's really with that knowledge base, we can then go into the lab and use synthetic biology tools to precision engineer gut bacteria to improve cancer outcomes.
So far we have cancer immunology, microbiome work, and artificial intelligence. So we're getting pretty fancy already this morning. Adam, will you please introduce yourself too?
Sure, yeah. Hi, everyone. I'm Adamyala. So as Barbara mentioned, I'm a PhD candidate at MIT, and I'm primarily a computer scientist. One of my primary application areas is how do we use the most widely available, widely collected data we have in the healthcare system, like patient imaging or disease codes, or like the kind of large-scale data we already have to predict relevant cancer questions. Who's going to get cancer? What is cancer risk? Who's supposed to respond to a particular treatment? How do we personalize treatments? And to develop machine learning tools to make these problems tractable and push the boundary of what we can do today.
Thanks, Adam. Zora, you're up next.
Sure, yeah. Hello, everyone, and thanks for the introduction, Barbara. So I am a computer scientist too. I'm based in Cambridge in computer science department, but we work very closely with oncologists from Cambridge as well. Our project is funded by Mark Foundation, actually a US-based charity, and we mainly work on cancer data integration. And one of our main aim is that to integrate data and actually bring it to clinical decision support system that can be used by clinicians on day-to-day basis. But using machine learning and bringing it to day-to-day practice is not the easiest thing to do. So there are a lot of challenges around that I'll be talking about during the discussion. Thanks
so much, Zora. So you can see from our panel, we have a lot of different perspectives and expertise. What we thought we would discuss today is the convergence of AI with emerging cancer therapies and kind of the innovation in cancer immunology and how that might meet AI and, as Adam said, allow us how to find out who's going to get cancer, why they get cancer, and then what we can do about it. So there's an enormous potential to apply AI to the challenges that we continue seeing, again, in studying, diagnosing, and treating cancer. Cancer biology has taken an incredible leap forward with immuno-oncology. But as we understand more and more of the biology, certainly the researchers who we work with at Alliance for Cancer Gene Therapy, the more we learn, the more questions there are, and the more data we create. So how can we bring all of that together and use AI and machine learning and all of these amazing tools to find some pattern and recognize the paths forward so we can diagnose more quickly and effectively and develop better therapies. So maybe we start with the study piece, the research piece. We have almost overwhelming possibilities with AI. If we can do anything, if we have the data and the information to do anything, how can we be intelligent about it? What are the problems that we want to solve, and then how we can solve them? So maybe, John, we can start with you. Just give us an idea of what's going on in immuno-oncology and where we've gotten to in that field, but also what are the major challenges where you think AI might be of help?
Yeah, all right, great. Thanks, Barbara. Yeah, so I can just get a little sort of a context in terms of what is actually, when Barbara says immuno-oncology or immunotherapy, what are we talking about, right? So, of course, traditional cancer patient would get chemotherapy, right? And that's basically, those are drugs that directly are cytotoxic to tumor cells. In contrast, immunotherapy is actually using drugs that turn on the immune system. They mobilize the immune system. That's actually your immune cells that end up attacking the tumor cells. And, kind of the major players, at least as far as as most of immunotherapy is concerned today, are so-called T cells. So their T cells have evolved. They're very powerful immune cells, and they basically surveil your body, right, and look for foreign stuff, right? In terms of evolution, they've mostly evolved to recognize virally infected cells, and they're tremendously powerful at eradicating those virally infected cells. But it also turns out T cells seem to do a pretty good job of recognizing that cancer cells are foreign, they can see that there's foreign material in cancer cells. That's because cancer cells actually mutate their genes and they start to look different than our normal stuff. But that posed kind of a conundrum, because there was, immunotherapy used to be developing tumor vaccines that would, specifically activate T cells against these tumor cells, and basically that was an utter failure. And so the huge breakthrough that happened was kind of in the early to mid 90s, the discovery of these so-called checkpoint receptors. Right? And so these are receptors on T cells. They're built into the immune system as sort of a negative feedback loop to prevent our T cells from basically attacking the body. And so, and tumors have basically learned to usurp that negative feedback loop and fool T cells into thinking that there's not a problem here, right? So you can think of these checkpoints as brakes on the T cells. They just stop them from doing their thing. And so, of course, once you discover these receptors, then you can actually develop antibodies that stop those brakes. They stick to those receptors. They prevent them from talking to the tumor cells. And you basically just, now really nicely activate the T cells. And, I think it was 2 years ago, the Nobel Prize was awarded to Jim Allison and Hanjo from Tokyo, basically, who had discovered two of these major checkpoint receptors, one called CTLA-4, one called PD-1. Right? And so the bottom line is this led to a breakthrough in terms of cancer therapy. There are other therapies, besides these checkpoint blockers. There are T-cell therapies called CAR-T. There are also bispecific antibodies that we utilize that specifically recruit T cells to attack tumor cells. So that all sounds great, but here's the problem. Basically, about 20 to 50% of patients respond to these immunotherapies, and we're not sure why. We have a lot of hints, right? There's rapidly evolving biology, trying to understand. Okay, there are other immunosuppressive factors. There are stromal cells that prevent T cells from getting into tumors. There are these MDSCs, they're myeloid-derived suppressor cells, there are regulatory T cells. All of these things can influence whether or not these immunotherapies will actually work. And so, the question then becomes, how do we figure out what's going on? And even more importantly, how do we know which patients are more likely to respond to these checkpoint therapies? And are there other therapies we should be developing that can complement the checkpoint therapies and help these patients respond that aren't responding to checkpoint therapies? Now, I'm not an AI expert. I'm not even an AI beginner, really. But it's not hard to think about applications of AI to solving this problem, right? So, again, why do some of the patients not respond? There are a lot of there are companies and organizations out there using AI as applied to image analysis. You take biopsies from these patients. You apply certain antibody reactions to them, immunohistochemistry, or just microscopy, or just CT scans, and these organizations are trying to use that data to inform whether or not these patients might actually respond to immunotherapy. There's applications that we use every day, where basically you get tons and tons of genetic data from tumors. And humans just don't have the capability to take 18,000 genes and figure out actually what's going on, and to use these machine learning algorithms that now reduce dimensionality in a way that they put them on these little 2D plots. It's like, oh, those are all the T cells, those are the macrophages, those are the stromal cells, and then we can interrogate those in terms of their gene signatures and figure out, oh, maybe that gene is an actionable gene where we can develop a new immunotherapy against that. I know companies, we work with this company called Gritstone that they're using AI to actually predict, personalized tumor vaccines. They take tumor genetic data, they figure out, how, mutations in those genes might relate to the immune system, and then they basically dial up a personalized vaccine for that patient. And then, last, of course, there's just general trying to relate bulk tumor genetic data to what the immune environment looks like, and does the genetic signature of the tumor actually influence the immune environment, and is that something we can utilize to figure out who's going to respond to these therapies? So those are just some examples, and I'm sure my colleagues on the call have much more sophisticated things to say about AI as applied to immunotherapy.
So John, you bring up that really big question about how are we figuring out what questions to ask, and with cancer research, we keep identifying knowledge gaps where we study and we research and we learn more and more about TILs and macrophage and how to understand what all of these various cell types and pathways are responsible for, and also how the tumor manipulates and is very intelligent in recruiting biology, your own body, for its purposes. So we have seen the exponential growth of available data, certainly in the last decade. But do we know how to handle it? So do we have the tools and the ability to take all of those different data sets and apply them intelligently? So maybe as a group, and I should say that if you want to submit questions from the audience, please do so in the chat. But as a group, we can have a conversation about the data itself and the multiple sources. And we have genetic data, genomic data, phenotypic data. Stephanie, you have all of the data from the microbiome. But how are we actually making it useful? So maybe, Stephanie, we could start with you and just tell us about data.
Absolutely. So we have some challenges in our space that are, I think, frankly, unique to the microbiome space. We don't have an infrastructure. There's no infrastructure for, nationwide collection of stool samples where we can truly understand what's happening in the microbiome, and especially in the context of oncology. And so because of that, we can't call up and work with a hospital on stool collection. It's not standard. So we had to create a standardized kit for collection of that. But what's important about the data, and it's very cliche to say garbage in, garbage out, but from the very beginning, when you're thinking, okay, we need to apply machine learning because our data sets are quite complex. These data sets, all of the genomics data sets, gigabytes of data per person, and it can add up quite, a significant amount of data quite quickly when you're looking at hundreds of patients or thousands of patients, for example. And so I think what we have learned is that we have to actually design our studies very carefully to make sure that, one, all of the samples that we're acquiring to do all of these genomics analyses are being, kind of standardized sampling and handling of those specimens, and then even how we then process that data has to be in a very standardized manner from the get-go, so that things can be reproducible and that we can get something out of machine learning. And I think what we've also learned in our space in particular is that data from hundreds of patients isn't sufficient to really get to actionable insights, something that is quantitative, that might be predictor of cancer. You need thousands of patients. And I think just the magnitude is significant, as well as really upfront when you're trying to design a study, you have to be extremely mindful of the type of data that you're getting, and if that data can be, put into machine learning algorithms. I think that you have to do a lot of upfront work, and I think that's been kind of the key learning that we've had.
Thanks, Stephanie. So Adam, I think you've had some of the same challenges in the work that you're doing in breast cancer, just even finding the data in the first place, or getting those samples that you need to before you even start your work. Could you speak to that for us?
Yeah, I think there's a the amount of healthcare data that exists is immense. The amount of healthcare data that is usable is much, much smaller. And I think the level of that gap is generally underappreciated. For a lot of things we care about, let's say we want to predict response to PD-1 checkpoint inhibitors from CT scans. Many people across the country have CT scans, have had the therapy, and have response data, yes or no. So in principle, we should be able to build tools to maximally try to predict this given the information we know about the patient. Now, in reality, it comes out that in many cases, we don't actually have the response data in any kind of categorical field. And so then to say, I want to understand this for my hospital, that would require doctors literally reading medical records by hand, and you just can't get sample sizes enough to learn something meaningful, that kind of thing. When you actually want to say, okay, well, there's big CT scan databases online. Do any of them have associated outcomes data of this kind? There are none. And so in general, there's this kind of big gap between what is conceptually possible and what is happening within the broader machine learning community and the kind of algorithmic tools we've developed at scale versus what's actually accessible for research purposes within our community in the United States in general. There's different kind of systems have had made different strides to kind of bridge this gap. The UK Biobank is very famous and kind of well recognized for trying to pull together these kind of data sets. Some of them, but not the kind of full picture to facilitate research. I think we have a long way to go within our general system. Because even like, mammography is incredibly common and hugely prevalent across the United States. But pulling together these resources to actually develop tools that benefits the entire patient population is incredibly difficult due to challenges of just kind of pulling together collaborations and data sets and just resolving kind of almost political issues that come into bringing together these resources.
So our lack of one system or our silos across the healthcare industry, which you hear discussed in larger, more general panels around healthcare policy and how do we actually run healthcare. That is in fact going to be a challenge for us when we come to applying AI and usable data to get where we want to be, where we can much more quickly and effectively use all of that data to advance therapies for patients. So, Zora, that is a really nice introduction for you to tell us something about how you're tackling that in your work in the UK, where we have a different system, and you have the opportunity to work with oncologists and pathologists who are all actually connected through the National Health Service. So maybe you could give us an overview of how that works outside the US.
So yeah, definitely. I mean, having a national health system, that definitely helps. And kind of that helps you to be able to enforce certain standards when collecting the data. But often one thing is to try to manage the production of the data such that it can lend itself better to being modeled by machine learning models. And it's another thing when you are given data sets already, that there's been some trials, they can go on for 5 years, 10 years, and then we get a data set from oncologists, they say, we've collected this over 10 years, what can you do with this? And as Stephanie said, interestingly enough, actually, often you have a lot of features for the same patient. So you might have 80,000 gene expression and mutation for a single patient, but you might have it only for 100 patients. And often machine learning algorithms are quite data hungry. And in that case, it's not possible to actually go back to trial and increase the data or anything along those lines, whether it be a national health system or a distributed one. So what I like to point out about this is basically the technical ways that we are trying to deal with this. And I have two particular one in mind that we are currently trying out. One of them is allowing the algorithms and models that are not as data hungry. And the other one is actually generating synthetic data. For For algorithms, so it's been shown that even the normal algorithms, by normal standard algorithms that we use, if we can try to bias them or put attention mechanisms on them, they can actually converge even in very low data regime. And we have done studies on that. So there are ways that instead of focusing on the data, there are ways of thinking out of the box and thinking of how can I make sure that my algorithm is actually still effective when I only deal with 100 samples and this huge amount of data for each specific sample. So that's one thing that I don't think it has been paid enough attention, maybe because this problem isn't particularly unique to healthcare and oncology. And the other one is generating data in ways rather than running trials and rather than actually finding physical patients out there. Again, so I think generative methods such as GANs and generative adversarial networks, they have been very, very successful in generating synthetic data. So basically, they observe the distribution of the real data that we have already, and based on that, they can generate data that is very similar to existing one, yet different enough for the algorithm to train on. And unfortunately, they haven't been used much in oncology, but they have been used extensively in image recognition, and they have shown to be very promising. So this is another thing that we are working on at the moment, trying to kind of increase our data set size by using generative methods. So hopefully things like that can help with the problem of the inequality of data sets without actually needing to go back to clinic.
I love that. So, you know, if we have a data hungry system, how can we find a way to feed it less, but still have it perform? It's really interesting to think about how we might apply that kind of thinking to the U.S. Because we're never going to fix the healthcare system in the U.S. We're just too big of a country, really, where every system is connected and data can flow, and we are all using the same standards. I mean, personally, I don't see how we get there. So can we be more intelligent about finding ways to use the data that we do have in really innovative and creative ways, particularly in cancer, because we don't have the time to wait until the system is perfect. And also trying to get patient samples, you know, and before we get the data, just to get the samples from patients is also another large challenge. But I don't want to take up all of our time just talking about the challenge before we even get started on what we can do once we have the usable data. So maybe we can move on and think about diagnosis, and as the first step for a patient in their cancer journey. How can we more effectively and more quickly diagnose patients? And certainly looking at something like pancreatic cancer, you know, right now we usually are diagnosing a patient when they're already at stage 4, and the outcomes clearly are extremely poor if your starting point is your cancer has gone really far already. So how can we address problems like that through screening and risk modeling? And maybe, Adam, we could start with you. You said that AI can help us ask better questions. So how do we do that to get us to a diagnosis?
Yeah, so I think a lot of what we view currently as guidelines are really computational problems, and framing them as such gives us a lot of tools that we can apply to them. So let's imagine breast cancer screening. We know that if you're above a certain age, usually the guidelines are like 40 or 50, you're now at high enough risk such that breast cancer screening is cost effective. So in this case, age is acting as the risk model. And we have an analogy of this in different diseases. So lung cancer, we say if you're over 50 and then you have 30 pack years of smoking. Now for pancreatic cancer, we don't have one. And so there is no screening. We have almost everything is caught at stage 4. But when we think about how do we find a population high enough risk such that screening makes sense for them, is cost effective, and the benefit to outcomes is not, this is the question of how we develop a risk model given the data that's available to us. So viewing it as this kind of computational problem, where we're not trying to presuppose, is it going to be age, is it going to be smoking? Now we can say, okay, well, let's take all the data that's available and just try to learn this function accordingly to try to identify which population high enough risk to benefit from screening. Once you start screening, now you're in a situation like breast cancer, we already have mammograms. The question is, now you have very rich information about the patient that's more disease specific, i.e. like an actual CT scan or an endoscopic MRI or an ultrasound, anything of that sort. That gives you further rich information yet to give you the ability to do better risk assessment. So one of the things that we've seen in breast cancer is if you take a woman's mammogram, you can predict risk much better than the wide variety of questionnaires like family history and whether or not the person has BRCA and like whether they have hormone replacement therapy. From the mammogram, you can predict risk much better. Given this information, now you can say, okay, well, how do I dynamically and optimally decide when to screen them? To say, okay, right now they don't have cancer, but they're high risk to get cancer within the next 3 to 4 years. Let's change the screening schedule in order to catch it at an earlier time point. And so from like the very beginning question of who gets screened, this is a risk estimation problem, which means we can use the most powerful techniques we have in the broader computational community to address it. From when you have screening, how do I actually change that guideline? Every year is not the upper bound of how well we can do, and how, we can do better by personalizing the resources to people's actual dynamic removing risk. And then once you actually have diagnosis, you can still build better diagnosis systems. So I think in every kind of stage of this pipeline, who gets screened, when did they get screened, and how did they get screened, you can build better tools and then get early detection along the way.
And Zora, you had also been talking about this. How do we keep enriching the data? How do we kind of treat the data set for each patient as part of their living system, if you like? Where all of the clinical data and imaging data and genomic data is coming in also in breast cancer. And I think one of the results that you had then also was a predictive tool that oncologists and clinicians can actually use in real time. Can you tell us a little about that?
So exactly as Adam says, it's very true that for certain targets, so if you want to see the likelihood of the risk of this patient being diagnosed with cancer in 2 years or 3 years, for certain targets, you might know mammogram is what performs best. So you can work your way backward. But one of the things we're dealing with, it's actually building a model of the patient. And that's based on every single bit of data that you have about the patient. So it's not about any specific target yet. So we have clinical data about the patients. We have genomic data, which could be expression, mutation, etc. And then we have the imaging data, mammogram, MRI, and so on. So at the very first stage, we kind of integrate all of this data and use that as a space, as a model to then try to do survival analysis and treatment planning for the patients. Something that we've noticed, what we've been informed by oncologists that happens a lot in their day-to-day practice, is the fact that in current standard of care system, when they do survival analysis for a patient based on only clinical data and imaging data, these two patients might have very similar survival outcome and might be recommended the same treatment. Yet in reality, what happens is that they are exposed to the same treatment and they get very, very different responses. One of them attains PCR, the other one does much, much worse. So of course, we know that the clues are in the kind of genetic background of this, and this is the factor that actually make precision or personalized treatment and survival analysis for the patients. But currently, there are not that it's zero, but the amount of genetic information that we use for survival analysis and treatment compared to what we are collecting, it's very, very little. There are known biomarkers, as Adam said, such as BRCA or estrogen receptors and so on that we use, but it's nothing compared to the whole genome sequence and that we actually might have about some patients. So at the very first stage, we make this integration and then we use that as a model or as a target of the patient for mimicking the response to treatment and doing survival analysis. And kind of that has the added benefit that then in a split of a second, an oncologist can expose this kind of model of the patient to many different treatments. So rather than at the moment they are growing, say, avatars, mice, to kind of grow along the life cycle of the patient, because if the patient is exposed to a single treatment, it hasn't worked out, what's the next one? So they want to know ahead of the time. But even our ability to do survival analysis and treatment and planning with avatars is very limited because they are very resource intensive. So kind of having that model that can be simulated for response to treatment, then clinician in the clinic, they can use this clinical decision support system, they can mimic the response, they can use that model for surrogate response and try many different treatment and think of what could be the best to expose the patient. So that's the main benefit we are after, creating this model of the patients to begin with as much as data that we have.
So I want to add one thing to that. There's kind of like two orthogonal directions of benefit. So when you add new sources of data, you can get huge, substantial boost because this alternate source of data can better explain the variance that's not explained by the original source. But it's not only adding more types of data, the type of modeling you do within the same data set can also make a transformational difference. So mammography has been used for risk modeling since like 1969. People use breast density, it's federally mandated, it's a human design biomarker that's not very predictive. We had some work like a year and a half ago that had one deep learning model that cast the problem a bit differently, tried to learn the biomarker and had a much higher performance. Since then, we improved it again. There's a better algorithm, and now there's a similar size gap between the old model and the new model. And so it's not only the sense of which data sets do I have, but there's a lot of room for just technical algorithmic innovation. How do we best capture the complexities of that data to do better prediction processes? Which I think is really exciting because then we've kind of transformed the like scientific hypothesis problem of like, let me imagine the right biomarker and find the right piece to say, okay, here was a signal, and now I can convert finding the right biomarker into an optimization problem. So all the kind of benefits in the broader community of how we develop better algorithms to understand images can then start benefiting the specific biomarker problem that we're tackling. And so there's kind of two independent thoughts, like how do we, like which additional source of data do we use? How do we better leverage their relationships, which is one learning problem in itself? And then per modality, how do we build better algorithms in general to handle this kind of tool? And this can also make transformational differences.
Yeah, absolutely. Yeah, agreed. I mean, the most kind of useful recent models, they use Ensemble, almost all of them. So the very recent work that was published earlier this year in Nature on breast cancer was this system that uses 3 different deep learning models and aggregates the prediction of these ones. Even in real life, if we can get 10 experts to kind of look at a pathology image and give a cancer score, of course, we have more likelihood of getting the right prediction. But with computation, you can just do the same. So you can use Ensemble of Models and then look at the aggregate of what they're suggesting. And that's a very useful thing to have.
It's amazing. I wish Zora and Adam, you could just go off now and stay in a room until both of you work together and come up with these incredible predictive models for patients. And so the thought that as a patient, before I have any treatment, that I could be informed with my oncologists on how I might respond to all of those various treatments, so that I don't begin a treatment that then locks me out of other treatments or clinical trials as I go forward. So the thought that we could have that knowledge as patients before we start our cancer journey would be make such an incredible difference than where we are today. So Stephanie, I wanted to bring you in here because I think the microbiome, we're getting some questions coming in, how on earth does the microbiome relate to cancer? Like how are those two things working together? So maybe you can dig into that. Tell us everything we need to know about our gut and how that might translate into better cancer treatments.
Absolutely, thank you. So yeah, I think most people don't recognize that the gut is actually the largest immune organ. And so the microbiome and its health, the products, the metabolic products from the microbiome have an incredible impact on our immune health in general, even from before we're born, in how it trains our immune system. It's really there to prevent and fight disease when it happens. And a lot of the work that we're doing in the space of microbiome and oncology stems off of 3 seminal science papers, back-to-back science papers in early 2018, linking in patients the composition of the microbiome. So the kinds of microbiome that are in these patients receiving checkpoint inhibitors for a variety of indications, that the composition is very much linked to the response. And over the last few years, what we've seen is that that continues to be the case. And now what we're seeing is that there's a tumor microbiome, and that tumor microbiome is also reflecting response to these drugs, largely stemming from aspects of gut barrier integrity and these microbes leaking from the microbiome and getting into these tumors and actually having an impact on local chemo, for example. But at a high level, what this is stemming from is that in those papers, and what we have subsequently seen in our own patient data, is that non-responders to checkpoint therapy have what we call dysbiotic or severely damaged microbiomes. They are missing key metabolic functions. They are inhabited by largely inflammatory microbial species, those that have actually been shown to cause cancer, and furthermore, pathogens. That's what's in their microbiome, a very inflamed state. And this is absolutely in contrast to the cancer patients that respond quite effectively. Most these super responders have microbiomes that look almost identical to a healthy person, rich in metabolic function, high diversity of the microbes. And so this has been shown in mouse models of cancer to actually have a causal impact. So similar to what Zora was talking about with avatars, we can do these avatar mouse models where we do a transplant of fecal material from cancer patients that are non-responders or responders, and we see the same phenotype in mice when treated with checkpoint therapy. So bringing together these real-world patient studies in combination to this causal impact, that's how we've been effectively able to link the gut microbiome and its impact on cancer therapies. And as John mentioned earlier, we're getting 20 to 50% at best response. And for many of these solid tumor malignancies, we're getting much less than 20% of response. And microbiome is very much a key component. Is it 50% of that aspect? Perhaps, but it is a dominant aspect here. And so what we're doing at Persephone is really trying to find the core metabolic functions that are missing in these cancer patients that don't respond, and to develop engineered microbial therapies that can be, as used as a monotherapy. We've seen that aspect in mouse models of cancer, but also in combination, as John mentioned, with immunotherapy in general, not just checkpoint inhibitors, but potentially CAR-T and immunotherapy in general, since the microbiome has very significant impact on immune response.
Stephanie, I wanted to ask you. I think there was a paper that came out, or it was a conference presentation showing that patients on pembrolizumab, that's one of the that's Keytruda, that's the drug that Jimmy Carter got. It's a checkpoint blocker. If they had been on antibiotics within 6 months of starting of therapy, they were far less likely to respond. And I'm curious if you can speculate on that.
Absolutely. And that initial observation of antibiotics and depletion of the microbiome was how people started investigating the role of the microbiome in impacting checkpoint inhibitors in patients. It was initially observed in mice, maybe about 5-6 years ago, that mice treated with antibiotics completely wiping out their microbiome, can't even detect any microbial species, lost the ability to respond to checkpoint therapy. And so at that point, it really demonstrated that necessity of having these microbes to mount an effective immune response to the therapy. And now we've subsequently seen it in multiple patient populations. So absolutely. And in fact, in our own data set, we're constantly monitoring antibiotic use by these patients, and we do see that impact as well.
I think there's a lot of people in the audience wondering what should they be eating, which probiotic should they be taking? And maybe you could shed some light on those questions.
Absolutely. I'm all about, and what's important here, we have to think about where are we in the world. We're in the United States, the Western diet. The Western diet has a tremendous impact on our gut health. And in fact, our biggest problem is the lack of fiber. So the biggest thing you can do for your health and gut microbiome health is fiber, because that's really what essentially gives the prebiotics that feeds the gut microbiome. And in addition, probiotics in the US are not regulated. And the best way to get probiotics is through fermented foods like kefir, like kimchi, like tempeh. That's the best way to kind of feed your gut from that perspective. But yes, fiber, lots of fruits and vegetables, and fermented foods is really the key.
We have this trend, of course, for probiotics, probiotics in everything and with everything, but kind of jumped into that as a health fad with no understanding at all of what that is actually going to do to your body and how it responds to other things, including cancer therapy. Yeah, that in and of itself is a whole other panel.
Absolutely.
So we're getting fairly close to our wrap up, but I thought maybe we could spend some time at the end here thinking about, so all of this is starting to happen today. We're all working in our various different sectors to look at how we can improve cancer therapies and treatments. Hopefully we're all converging together and that in some part, due to COVID-19, we're learning to be a more collaborative group of researchers and companies. But let's think about in 5 years' time, is everything moving fast enough where in 5 years' time as a patient, I can walk into my doctor's office, have that spread of my living data available to me and the doctor. I'm not feeling great. This is what's going on. Okay, let's put that into you, virtual you, and let's look at what might be causing your discomfort. And getting really quickly to a prediction of an early cancer diagnosis. And then on the next screen, all of the available treatments and how they might work for me or not. How far away are we from that? 5 years, 10 years? What's your opinion, guess?
My gut tells me about 15 years, maybe. I don't think 15. It's an easy problem.
And it's your gut that's telling you. Stephanie's going to like that. So 15 years out, okay?
At least.
Yeah, 5 years is definitely a bit too good to be true.
I remember back in 2000, I think, where there were panels about, in 5 years we will cure everything because we have the genome sequencing. So that didn't happen. So maybe Stephanie, you can tell us where you think we'll be.
I really think around 10 to 15 years, but I think there's a tremendous amount of work that has to be done in our infrastructure and retooling it so that we can apply these data analyses very early on and collect the right data sets and have them in, almost central data warehouses. Right now, so much data is siloed and even within the individual physician's practice. So I think it's really an overhaul of the healthcare system from a data management perspective. And how we handle that data is really what's going to be critical to the next 10 to 15 years of having this moonshot, essentially.
I totally agree. There are some bottom-up approaches and some top-down. So the bottom-up approaches are doing really well. Say the startups and small companies, research-based companies, are doing a very great job in actually showing proof of concept and showing that these methods actually work. But there is definitely a huge amount of kind of top-down infrastructural efforts is required to make this a part of the health system. Even in the UK, that there is a national health system, there are all kind of policies that need to be put in place. There is things outside immediate research that we are doing, such as the privacy of the patients. And there are a lot of things that need to be taken into consideration and acted upon to make this a reality.
So I think there's some things we can accomplish in 5 years. Like, we have some tools that can help advance the standard of care today, but whether or not those things actually, like the gap between technology existing and it actually benefiting patients, depends a lot on what we as stakeholders all do within that time frame. So I think it's quite possible that we get some of the benefits within the next 5 years, but obviously there's always going to be a long spectrum of how hard each individual problem is. I think that we have really great systems and kind of regulations in place to say patient data should not be just given away, and we need to have like strong protections around medical privacy. I think we don't have a kind of as well established system, at least in the US, of patient data is a national resource and patients globally benefit if we can make it available for people to develop new tools from. And I think both in terms of even existing models, how quickly they can be validated, how quickly we can develop policies using them, all of that is conditional upon how much progress as scientists in terms of how well we develop the tools, but also as the public saying, should the data be held in one hospital as a part of a larger public trust and be accessible for like general public research? We have some resources towards this direction, things like the NIH resources and the big clinical trial are really great and useful and has developed a lot of new tools for lung cancer in terms of like automated detection. But I think as the public and as stakeholders, the timeline is not this autonomous process, and it's something we can influence by trying to make the system better. Systemically, for now, things are slow and it's an uphill battle, but we can do better.
And it's amazing to me that, I can walk around with my iPhone and I'm generating constantly so much information about myself, regardless of my opinion on privacy. I'm somehow not connecting those two things where I don't want anybody seeing blah, 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 blah. Meanwhile, on my phone, I'm sending out that information constantly to the world at large. And it is being used to, as Stephanie brought up at the very beginning, if I go on to Amazon, it's going to start telling me like what I should be doing from a purchasing perspective. Somehow we know more about me as a person, as a shopper, than I know about me as a person, as a healthy or unhealthy individual. So, is there a mindset that needs to change around privacy, for example, that does allow us to share data more freely, or does that get us into a difficult place where that data can be used without our knowledge or without for an intent that we don't necessarily agree with?
There is a lot of exciting work in even the current generation of kind of like consumer technologies. There's a lot of effort in making algorithms that preserve privacy while still be able to learn the functions to be useful. I think there's large opportunities for that. And this field is, there's like the homomorphic encryption side to say, can you train models using encrypted data, do inference on encrypted data? And there's a lot of great progress. I think is going to make the problem continually easier. But at the very base level, for that to succeed, there needs to be kind of a will to, we need to solve this problem collectively in kind of like larger cross-institutional collaborations to facilitate those kind of approaches gaining traction. I do think that I don't envision a world in which the optimal we find is everyone could look up everybody else's like health records. That would be some nonsense. But something we can't do today that would still be private, but we should be able to do, is if I walk into a hospital, I have some disease, I have some comorbidities, I have some prior history. If I want to say, how many people fit my profile in this hospital? As of now, this is not a question you can ask beyond the most basic thing. Like, how many people had these symptoms, had this disease, got this treatment in this hospital, what happened to them? These are like very basic, like, you can say how many people bought an iPhone on like March. You can do lots of kind of very basic analysis. It's not even like fancy machine learning analysis. And we can't answer that now. And so some of that, and some of that is building better tooling, and some of that is supporting is, okay, well, if not enough data in the hospital, can I go across my state? How much, like, I think we don't think enough of our data is instead of a property of only the hospital or a single clinician you're working with as a national resource that we can all benefit from.
And so John and Stephanie, as two CEOs of biotech companies, do you think we'll get to a place where within the amazing innovation of biotech in the US, where companies will more freely share what they're doing for the greater good, or is that just, again, never going to happen? Does that go against the model of biotech in the US?
Boy, yeah, that's a tough one. I'm not a CEO. I'm a CSO, which is a much better job. Yeah, I mean, there's intellectual property, right? Everybody's worried about somebody else using their information to come up with a better therapy or so forth. But it seems ironic to me that because we've been trying to cure cancer for decades and haven't been able to do that. But when COVID hit, there was a consortium amongst all these major pharmaceutical companies saying, "Hey, we're all gonna work together. We're gonna share data. We're gonna share assays." And they actually there was this really great altruistic sense of like, wow, we got to work together to solve this quickly. So I don't want to rule it out, but the intellectual property factor, it has to be worked out, I think, in a way that people can share data, but IP is still protected.
And to add to that, I think is the best place for actually this collaborative effort, and the Parker Institute of Cancer Immunotherapy is doing that. As a nonprofit, bringing together both academic institutions with pharma companies and small biotechs, and they're making it work quite successfully. And I think it's that model that we need to learn from. We're also a partner of theirs, and I think it's that type of partnership that's really critical, especially in oncology, to John's point. There are many times where we are trying to approach various institutions to work with them, but they already have conflicts of interest and they can't. But we're trying to partner on the same thing. We're all on this, fighting the same cause. We need to be better about that, and we need more infrastructure towards that.
So as we're also a partner with the Parker Institute and the Cancer Research Institute, focused on cell therapy and glioblastoma, because glioblastoma is a situation where getting the samples from the patients and getting enough samples from the patients is very difficult, and the only way forward is to get all of those samples in one place and analyze them in the same way, so that the available data is of a quality where the cell therapy research can happen. But the output from that part of the collaboration is that the resulting early IP would then be licensed out to the biotech companies. So it's really an engine to drive work forward in glioblastoma that then industry can pick up. And IP, of course, IP is kind of broadly applied to everything that you've ever developed within that company, but some of it you're not going to do anything with, right? So like let's set that data free, and don't hold on to it just because it was a great idea at one point. But again, I think we're straying into another panel. So I believe our time is getting close to being up. It's a 9:15 wrap, yeah? But I just want to look at questions that have come in. We have some comments, really, rather than questions from the audience, about, Stephanie, the microbiome, which can be unique, and requirements for exome analysis. An example in IBD, intestinal bowel disease. can we easily pivot data outputs from the analysis towards association studies from similar approaches? Any thoughts on the microbiome and IBD?
I mean, absolutely. A lot of the seminal work in the gut microbiome space that actually led to many of these insights in oncology was from the IBD space, because there are a lot of genome-wide associated studies initially conducted in IBD. But there are, I think these kinds of analyses have to be applied specifically to various diseases. There are commonalities, there are microbial functions that are in common, but each of these diseases seem to have a unique framework. So I would say there is something that we can leverage. Obviously, just more data, the better that we're going to get at understanding maybe some of these commonalities, that maybe similar drugs could be used for both illnesses.
Okay, so we are indeed at our wrap-up time. Anyone have a closing comment they'd like to add? Personally, for me, I'd love to just keep all of you working together on curing cancer, but we've got to let you get back to doing that in your day jobs. Okay, well, thank you, everyone. I hope that this panel's been helpful to the audience, and we'll see you next time.
Thanks for having us.